Welcome my colleagues and my dear students. We are speaking about the resting membrane potential. Actually, we have two important related topics regarding the membrane potential. First, the resting membrane potential, or sometimes we call it resting potential. The second is the action potential or the nerve impulse. Both of them are related to each other. We can't obtain action potential, which is the secret of life. As we know, everything in life, in biology, is dependent on nerve impulses to see, to hear, to move, to digest, to respire, for the circulation, for the heart. Everything in life is dependent on the nerve impulses or the action potential. To have action potential, we must have resting membrane potential. The relation between them are so close, like the relation between the sea and the wave. We must have the sea all of the time. Waves may come slowly, may come rapidly, may not come at all. Also, we must have resting membrane potential over the cell membrane all the time. Impulses may come rapidly, slowly, may come repeated, or may not come at all. This is the resting membrane potential or the resting state or the polarized state, where the resting potential is present all the time. However, the nerve impulse come as a wave of depolarization. It may be repeated rapidly, slowly, or it may not come. So what is the definition of the resting membrane potential? The resting membrane potential is the potential difference between the outside and the inside of the neuron in the resting state. One microelectrode outside, the other inside. There is a potential difference of about minus 70 millivolts. The value of the potential may differ from a neuron to another. The value of resting membrane potential, as I said, in average about minus 70 millivolt. It is 70 millivolt and we call it minus 70. Why? Because by convention we put the charge of the inside of the cell in front of the value of the potential. So when we say the potential difference is minus 70 millivolt, this means that the difference is 70 millivolt and the inside is negative and the outside is positive. This is the unstimulated state in biology. We must ask ourselves two important questions. First, why neurons are polarized? The second, why the positive pole is outside and the negative inside? The answer is in this topic, which is the causes of resting membrane potential. We have three causes. The most important of them is selective permeability. It is the main cause of resting membrane potential and it is responsible for about 90% of the formation of the resting membrane potential. The second is also important, which is the sodium-potassium pump. The third, which is the unequal distribution of ion, is a result of the first and the second causes. So what is the selective permeability, which is the main cause of resting membrane potential? Our cell membranes, especially in the neurons, can allow diffusion of ions. However, the diffusion is selective, so that the channels which are present in the cell membrane proteins are designed or they are tailored for passage of certain ion. This is controlled by certain size, certain charge, and certain diameter. So what fits certain ion can't fit any other ion. This is a cell membrane and these are the channels. As I said, passage through them is selective for certain ions. This is simple idea which can answer the question why the neuron is polarized and why is the positive pole outside and the negative inside. Potassium is present inside by a concentration of about 140 milliequivalent per liter. Outside only 4 milliequivalent per liter, so that the concentration inside relative to outside is about 35 times. For the protein, its concentration inside is about 16 gram percent, outside 2 gram percent. So the concentration inside is about 8 times the concentration outside. According to the rule of concentration gradient, both potassium and protein tend to move outside. However, the cell membrane is impermeable to protein, so protein is not allowed to move outside. However, it allows diffusion of potassium through its specific channels. So inside the cell, we have positive, which is potassium, negative, which is protein. Potassium is allowed to diffuse outside and protein is left inside. So positive ions move outside and the negative protein is left inside. And this process is continuous for life and it creates a positive pole outside and the negative pole inside. This process is maintained by the sodium-potassium pump, which keeps very high concentration of potassium inside. So all the time potassium is higher inside than the outside, so it diffuses from inside to outside all of the time. This simple idea is the secret of human life because it is responsible for generation of the polarized state, which is the resting potential, which is the secret of action potential, which is responsible for everything, for every activity in our life. We have three basic types of channels in the cell membrane. First, the passive channels or the leakage channels, which are responsible for the resting membrane potential. The second is the chemical activated channels or the chemical gated channels, which are responsible for the neuromuscular transmission and the synaptic transmission. The third is the voltage gated channels, which are responsible for generation of nerve impulses. First, what is the idea of passive channel? Passive channels are called leakage channels or non-gated channels. Through them, ions can pass through the cell membrane all of the time and they are responsible. And while speaking about the potassium ions, the potassium diffuses from inside to outside through this type of a channel or a similar type of these channels, and this is responsible for creation of the resting membrane potential, but they have no role in the action potential. The second type is the chemical activated channels. They are gated channels. When the chemical transmitter is released, it binds to specific receptors so the channel opens and ions can pass through the cell membrane. This is the idea of the Keynes lock. It is present in the neuromuscular junction and it is responsible for neuromuscular transmission. However, it has no role in the resting membrane potential or the action potential. The third is the voltage gated channel, which opens and closes by voltage only. This gated channel opens and closes by voltage. It is responsible for the action potential, but they have no role in the resting membrane potential. Sodium-potassium pump, the second important cause of resting membrane potential, and its role is crucial for formation of the cell membrane, and they may work side by side to the selective permeability. Sodium-potassium pump work to build the concentration gradient differences which are essential in biology. The pump works to build a concentration gradient difference for sodium and potassium. The concentration of sodium outside about 140 milliequivalent per liter, so the concentration outside is 10 times the inside. Potassium is inside 140, outside 4 milliequivalent. The concentration gradient is about 35 times. These concentration gradients are so important in biology for membrane transport and for maintenance of resting state and also for the action potential, which will be discussed later on. To simplify the role of sodium-potassium pump, we can conclude it into 6 steps. The first step, it binds to 3 sodium inside of the cell. The second, the alpha subunit becomes phosphorylated. The third, it releases 3 sodium outside. The fourth, it binds to potassium from outside. Then dephosphorylate. And lastly, it releases the 2 potassium inside. Then the process repeats itself for life. The pump works all the time as long as ATP is provided. So what will occur if the pump is inhibited? As I said, the presence of the pump is essential for maintenance of resting state. It is needed side by side together with the selective permeability. If the pump is inhibited, the diffusing sodium inside through its passive channels will accumulate inside, and this will cause neutralization of the negativity of protein, so the negative pole is lost inside. At the same time, potassium will move away from the cell membrane because it is attracted to the negativity of the cell membrane. So if the sodium-potassium pump is inhibited, this causes loss of resting membrane potential, loss of excitability, and loss of action potential, and loss of life inside the cell. The third cause of resting membrane potential is unequal distribution of ion. This is the result of the effect of the selective permeability and the effect of sodium-potassium pump. So we can note easily that the sodium concentration outside is 10 times the concentration inside. Regarding potassium, the concentration inside about 35 times the concentration outside. Chloride is about 25 times the concentration of outside in relation to the inside. Also, protein is concentrated inside. These concentration differences are felt by the activity of pump together with the selective permeability. Thank you, and see you again in the lecture of action potential.